Hey there, Mr. Mechanic. I got another podcast for you. So this one deals with some basic chemistry on the pH scale. So yes, you're probably familiar with this pH scale, this acid and base scale that measures the... Wait, what does it measure? Well, that's what we're going to find out today. We're going to find out what the pH scale is in fact measuring. So when something has a pH value of 5, what does that actually tell us? Or if something has a pH of 12, what does that actually represent? So that's the goal. Figure out what the pH actually measures when we talk about different solutions and why this is so important in biology. So let's get ready to talk about acids and bases.
Because you know I'm all about that bass, no trouble. I'm all about that bass, no trouble. I'm all about that bass, no trouble. I'm all about that bass.
So you might be thinking it's all about that base, but in fact, it has a lot to do with the acid. So what is pH measuring? That's the big question. So when we talk about pH, we're talking about the measurement of the hydrogen ions that are released into a solution. So the P is for the power, the H is for hydrogen ion. So what happens is if we have water, we know that water over time can dissociate. It doesn't happen to every water molecule, but some of these water molecules separate apart. And what's left over is this hydroxide ion, so a oxygen and a hydrogen ion bonded together, and what's released is a hydrogen ion, an H plus ion, which is nothing more than a proton. So when these accumulate in solution, it creates an acid. So if we have a surplus of hydrogen ions, more hydrogen ions than hydroxide ions, we have an acid. So an acid is nothing more than a substance, when dissolved in water, releases hydrogen ions or protons. So the extent at which molecules will dissociate depends on that substance. Some substances, like water, don't dissociate very easily. A substance like hydrochloric acid will fully dissociate and release lots of hydrogen ions into solution. So when we talk about acids and bases, we're going to be referring to this dissociation of different molecules. So sometimes we'll use the term hydroxide. Sometimes you might come across the term hydronium, which is this positive ion that actually collects one of these protons, and these can accumulate, creating an acid environment as well. So if we think about some solutions, like hydrochloric acid, it's made up of a hydrogen ion and a chlorine ion bonded together. So when these things enter into a solution, the hydrogen and the chlorine ions will fully dissociate. They will separate, creating a surplus of hydrogen ions, which creates an acidic solution. If you have a substance like sodium hydroxide, and the sodium and the hydroxide ions dissociate when they are in a solution, what happens now is you have a surplus of the OH. So now we're talking, that base. Right here, sodium hydroxide dissociating into hydroxide ions makes that base. So how can we actually calculate the base of a solution? Or how could we go about calculating what the pH is? Well, if we know what pH stands for, we know it's the concentration of hydrogen ions, we can define pH as the negative logarithm of the hydrogen ion concentration in a solution. So a logarithm is the power to which a number must be raised in order to get some other number. So for instance, if we use a base 10 logarithm of 100, Base 10 means that we can multiply everything by powers of 10. So a log of 100 is equal to 2, because if we raise 10 to the power of 2, 10 squared, that's equal to 100. So our pH equation that you guys will need to remember is this. The pH of a solution is equal to the negative log of the hydrogen ion concentration in that solution. First, we would have to know what the hydrogen ion concentration is. So very often this might be given to us in a problem. I might be able to tell you that this known concentration of our hydrochloric acid solution is 0.5 molar. So molar is a measurement of the concentration of moles, which is a specific number of atoms in a given quantity. So for those of you that take chemistry, you'll learn a lot about moles at some point in your chemistry class. Just right now, you should understand molarity is a unit that we use to describe the concentration. The brackets in this equation right here, these brackets always represent concentration. So pH is simply the negative log of the concentration of hydrogen ions that are in a solution. If we wanted to calculate the pOH, so pOH, that means the power of the hydroxide ions, which is equal to the negative log of the hydroxide concentration. So in all, if you took the sum of the hydrogen or the pH and the pOH, this must always equal 14. So if the concentration is equal to 14, and we know what the concentration of either the hydroxide concentration or the hydrogen ion concentration, we can determine one or the other variables, because both of these must be equal to 1 times 10 to the negative 14th. So we're going to take a look at some practice problems to give you an idea how we can calculate this. And to kind of conceptualize what I'm talking about, we can think about if you have a lot of hydrogen ions in a solution. This gives us a very, very low pH. If the entire solution was filled with hydrogen ions concentration, the maximum amount would be 10 to the 0 power, which gives us a pH of 0. So theoretically, this is possible. More than likely, solutions aren't going to be totally saturated with hydrogen ions. There's always going to be a small amount of hydroxide ions in that solution. But the more hydrogen ions there are, the lower the pH, because it's a negative log. If you have a lot of hydroxide ions in the solution, that means you have very few hydrogen ions. As a result, now we have a pH of 14, and we have a concentration of hydrogen ions equal to 10 to the negative 14th, which is a very, very small number because there's fewer of them. So if we know one or the other, the pH or the pOH, we can go ahead and calculate this. And why might we want to know this? Well, because most living cells function in a very specific pH range. Okay, so if the pH goes down for some reason, this could have a huge impact on cells. The proteins might not function properly, diffusion might not take place across the cell membrane, and it could really cause a cell to go through apoptosis and die. So when we talk about hydrogen ions, we're referring to this proton that can accumulate in cells or in living tissues and can have a huge impact on organisms. When we look at blood, for instance, blood has to remain within a very, very narrow pH range. So if you look here, we're looking at about 7.4 for a pH of blood. Blood has to remain very close to that, because if it doesn't, those proteins in your blood, the hemoglobin proteins, will malform and they are not going to function and transport the necessary oxygen to your cells. So if we think of milk and eating eggs or coffee or orange juice, all of these substances have different pH values. And when we digest them and absorb them into our small intestines and eventually into our bloodstream, all of these can have a huge impact on cellular functions. So again, small slight changes to a pH can have a huge impact on how these cells perform. So even though these numbers 14 through 0 don't seem that significant if we change from 7 to 8, well, if you think about it, every factor of 10 difference in the concentration represents 1 pH unit. So for every 2 difference, for every concentration of 2 difference, this represents a change in 0.3 pH units. So therefore, very small numerical differences in pH can have huge effects on biological systems. So one of the things that we're going to talk about and learn about is how we have certain materials in our cells that can help alleviate some of these changes by using what are called buffers. But we'll save that for a whole nother presentation. So let's think about what I mentioned earlier. If we know that the pH of a solution is equal to the negative log of the concentrations of the hydrogen ions, and we wanted to go through and do a calculation. So it says, what is the pH of an acid solution with a H plus concentration of 0.0001 m? So this is that molarity. So the concentration is very small. This is a very low concentration of hydrogen ions. So if we know that the concentration is equal to this number, this is also equivalent to 10 to the negative 4th power. So again, if we put this into scientific notation, it's 10 to the negative 4th. So you will need to do that for most pH calculations. So now, what you have to do is you have to use that little log feature on your calculator. So the calculator has a function that's called log. This will automatically take the 10 to the negative 4th and determine what that equals to. So if you plug in the log of 10 to the negative 4th in your calculator, you're going to get a negative 4. Well, we can't have a pH value that's negative. So if you remember, we said the pH is equal to the negative log of the hydrogen ion concentration. So if we did in the calculator now, we do a negative symbol, then your log, and then you might have to put this in parentheses depending on your calculator, you will get a positive 4. So the pH of a solution that has a concentration of hydrogen ions that's 0.0001 has a pH of 4. That's how you do it. That's how simple calculating pH can be. So we are going to spend some time doing a lot of calculations in class, making sure that all of you can calculate the pH correctly. So I have one more example. So if we know the concentration of the OH ions, so let's say we have sodium hydroxide as an example, and it has a concentration of 0.001 m. Well, if we wanted to find the pH, we have to keep in mind a couple things. We know that POH is the negative log of the hydrogen ion concentration, and this is what we know. So if we plug into our calculator the negative log of 0.001, or you could put in 10 to the negative 3rd power, you would get a pH of 3. Excuse me, a POH of 3. This is not the same as pH. So the POH tells us the POH of 3 must be the sum of the hydrogen, the pH and the POH must be equal to 14. So if we solve for pH, we simply have to take the 3 and subtract it from the 14, and we get a pH of 11. Just remember, anytime we are dealing with hydroxide concentration, which is going to be a base, we should have a pH greater than 7.1. Bases are always greater than 7.1. So here's what I would like you to do. Here are 4 practice problems. I would like you to do these in your notebook and come to class tomorrow with these answers. We will discuss them and we will go through to make sure that all of you are able to do